Which amino acid is the only common one that is not chiral? Glycine, alanine, serine, or leucine? Pick one. We will circle back after we talk about stereochemistry. In this video, you will learn the core structure of amino acids, how pH changes their charge, and how side chains fall into useful categories that drive protein structure. An amino acid has three key parts: an amino group, a carboxyl group, and a central alpha carbon that carries a variable side chain we call R. That one swap, the side chain, gives us variety across the 20 common amino acids. In water near physiological pH, most amino acids exist as a zwitterion. That means the amino end is protonated as ammonium, and the carboxyl end is deprotonated as carboxylate, giving one positive and one negative charge on the same molecule with a net of 0. As conditions become more acidic, extra acid can add a proton back to the carboxylate, yielding a net positive form. In a strongly basic solution, the base removes a proton from the ammonium, yielding a net negative form. Around neutral conditions, the zwitterion dominates. Now, side chains are where the action is. Some are hydrophobic and prefer the protein interior. Some are hydrophilic and reach for water. Some donate or accept protons and carry charge. Some are aromatic rings. Others are flexible and aliphatic. This diversity drives how proteins fold and function. Try one. Lysine has a basic side chain. In an acidic solution, many sites hold protons, so the net charge tends positive. Near neutral, the backbone sits in the zwitterion form, and the side chain can remain protonated. Far into basic conditions, the amino groups lose protons, so the net charge shifts toward negative. Here are two quick fixes. First, zwitterion does not mean no charges. It means both positive and negative charges are present, and the net is 0. Second, glycine is the exception that is not chiral because its side chain is just hydrogen. Let's do a mini checkpoint. You can now name the parts of an amino acid, explain the zwitterion, predict forms across acidic and basic conditions, and describe how side chains shape behavior. Ready to lock it in? Tap the link for study guides and flashcards on this topic, so you can keep learning without missing a beat. Time to talk about stereochemistry. Remember the question from earlier? All common amino acids have a chiral alpha carbon, except glycine, whose side chain is just a hydrogen. And in living organisms, proteins are built from the L amino acids, not the D set. Another useful lens is nutrition. Humans can synthesize 11 of the 20 amino acids, so those are called non-essential in the diet. The other 9 are essential, meaning we need to obtain them from food. There is a gray area because tyrosine can be produced from phenylalanine, which itself is essential. Let us apply the side chain categories. A long hydrocarbon tail is nonpolar and tends to tuck into the protein core. A side chain with an amide group is polar and comfortable at the surface where it can touch water. This simple filter helps you predict where residues like to live. Now, not every amino acid you meet is part of a protein. Some amino acids and amino acid derivatives act as signaling molecules, such as neurotransmitters and hormones, even though they are not built into proteins. Let us practice. First, sketch the zwitterion by placing a positive charge on the amino nitrogen and a negative charge on the carboxylate. Next, decide whether the environment is acidic or basic and adjust the form. Finally, classify the side chain by polarity or charge to predict behavior in water. Here's the big picture. All amino acids share the same backbone and usually appear as zwitterions in water. pH shifts push them toward net positive or net negative forms. Side chain categories explain folding and function. Biology uses the LSAT, and in humans, 9 amino acids are dietary essentials. Now that you've watched the video, let's see how much you remember. Head to the link in the description where you'll find Quizlet content made just for you.